Okay, thanks very much, David. So I'm not going to thank the organisers for inviting me. So we've heard a talk about sequencing individual genomes sort of with great accuracy, and we've had talks about sequencing some genomes in large numbers of individuals. I'm going to be putting those two axes together and talking about sequencing lots of individuals across the whole genome, and in particular the 1,000 Genomes Project, which many of you have heard of, which is very much an ongoing project which is aiming to sort of produce a very comprehensive catalogue of human genetic variation by sequencing what we call at the population scale. That's kind of a meaningless term, but it gives a sense that you're trying to sequence lots of individuals. So the types of things I want to talk about, the questions I want to address are, first of all, why do we actually need a comprehensive map of human genetic variation? What's it going to do for us? Very related to that, how is the data from the 1,000 Genome Project actually going to be used in medical genetics? And there I'm going to echo some of the things that David said earlier about screening individual genomes, but also looking into other ways in which you can use project data. And then finally, perhaps as a way of trying to sort of start some of the discussion later, what are the big challenges still lying ahead in sequencing at the population scale, lots and lots of genomes? What are the things that we need to worry about? So David put up a picture showing how Mark Henderson, the sort of, as he said, the best science correspondent in the UK at the moment, is very much on board with the idea of the genetic revolution, but it's not a sort of an opinion that's completely widely shared. Here's a headline from a paper at the opposite end of the scientific spectrum, where, as you can see, the headline is that there's a false hope in the hunt for genetic cures, and this is pretty much the same time as that Mark Henderson article that David showed, saying that there's a revolution here. And so, of course, where this is coming from is the idea that although genome-wide association studies and our genetic revolution has made a lot of progress in identifying bits of the genome associated with various phenotypes, whether it's disease or adverse drug response or so on, actually in terms of the predictive value of knowing an individual's genome sequence is still typically fairly low, except in a few cases. And so that's where there's this sort of the big open question is, how can we actually get down to being a bit more predictive? Is it maybe genetics is actually never going to get us that far? Well, that's one possibility, but over the last few years there have been a whole bunch of papers. Here's just a few I've selected more or less at random, which actually suggest that there's really a lot more in the genetics. We're just not getting at it yet, and that's either because we're not able to screen the very rarest variants that exist in a population, or we're not looking at all the variation, even the common variation, or that the effect sizes of many of these things may be really very small, so that you need thousands and thousands of individuals before you can really see statistically significant effects. So there's more to get out of the genetics, and actually probably a lot more to get out of the genetics. And so it's really worth putting more effort into it, at least that would be my argument. But how we're going to move forward from where we are to a deeper understanding of the genetics, well, what we need to be able to do is to assay genetic variation, not just at those few 100,000 or maybe up to 1 million SNPs that people are typically looking at the moment, but across the whole genome, so all your variation, and not just your SNP variation, all your different forms of variation, whether it's indels or structural variants or copy number changes and so on. And so what the 1,000 Genomes Project is doing is trying to provide a baseline for these studies. On the one hand, this is what variation looks like in the normal population, and by doing that, it's going to give you a reference that you can use in your own studies, and a reference by which you can compare your own sequenced individuals too. So the 1,000 Genomes Project is, at one level, simply a catalogue or a map of all types of genetic variation, including these rare variants And we aim to be quantitative about this goal in the project, so we're aiming for things at 1% frequency and above, obtained by sequencing at least 1,000 individuals, and it's looking more like 2,500 individuals at the moment, from geographic centres of major medical genetics interest. It's a big collaboration, of which I am a tiny part, across the world, including many of the companies that are actually developing these high-throughput sequencing technologies. And so we're really looking at the use of these high-throughput technologies for population-scale sequencing. So sequencing not just a few individuals, but a whole cohort. And of course, ultimately hoping to drive the rate of disease mechanism discovery. So this is what the samples for the project look like. They are deliberately unphenotyped. So David on David's axis, that was definitely a negative to not have clinical data. But remember, we're trying to provide a picture of sort of normal variation. We don't want to try and select for people with particular phenotypes. And in addition, all the individuals that we are sequencing, there are established cell lines available. So actually, there's a lot of information about molecular phenotypes, if not their say, disease phenotypes. And so the way in which the project is working is to take about 100 individuals from clusters of populations, related populations, about 100 individuals from each of these populations, spread from across the world. There's a European centre, there's a sort of Bantu-speaking Africa centre, there's an East Asian centre, a South Asian centre, which I haven't put the individual populations on, and then various populations from Central and South Americas. So that's what the project's looking like, and we would expect the data from this to be complete. Quite a lot of this will be done by the end of the year, but then maybe another year for the remainder. We haven't completed this, but what we have completed is sequencing of a few about 180 genomes from a series of pilot populations. So rather than launch into a whole project, we did a pilot to see how well our approach is going to work. And so what we've done is to sequence 60 people across the whole genome from those populations shown. The ones with stars, we've only sequenced exons for. We haven't sequenced the whole genome. So here's a cartoon showing you the structure of the pilot projects that we've carried out. What we have, there is a pointer somewhere. You're pointing. Ah, sorry, yes, being even dumber. Yeah, sorry. So what we've done is to sequence two trios, mother, father, child trios, one from Europe, one from Africa, to very high coverage. And so what the idea is showing is that these individuals have different haplotypes. The bars are meant to represent where there are genes. And so what we get from this is sort of very high quality genomes, individual haploid genomes where we phase by transmission from the parents to the trio. So essentially this is as good as it gets. We've done an exon-centric study, which is like the exome sequencing that Nigel talked about, where what we're doing is just pulling out the exons of genes, sequencing them to high coverage. And in these, you typically get very good genotypes on the exons, but it's hard to link them together because you don't have the sort of linkage to disequilibrium data typically. So these are typically unphased. And then the other type of study we've taken, and this is the sort of novelty in the project, is that we are sequencing most of these genomes, these 180 genomes, actually at very low coverage. That's between 2 and 4x coverage. Now I'll explain a little bit about why this totally stupid strategy actually works in a second, but what we hope to get out of that, what we can get out for that, are the sort of good estimates of common haplotypes. We have to use statistical methods to combine the information from these different individuals, and I'll say a little bit about how we do that. Okay, so that's the structure of the pilots. That's complete and all the data is available, and as of 2 weeks ago, there's even a paper submitted describing it. So just a bit about this population scale sequencing where you're actually doing low coverage on any one individual. Now it does sound like a bad idea because if I would just give you 2 or 4x coverage on a single genome, you wouldn't be able to do anything with it, basically. So here's the cartoon. You've got an individual. This individual carries 2 copies of each haploid genome, and haplotypes are typically different from each other. In this case, over a short interval, this individual carries, say, a light blue and a dark blue haplotype. These are carrying different variants, and they're sequenced, say, to 4x coverage. So when I sequence data from this individual, it's typically low coverage. I can't do much with it. But in the population, haplotypes, my haplotypes over a limited part of my genome are quite likely, or very likely, to be similar to other people in this room. And so over a sort of, by the time I'm looking over a large number of individuals, any one haplotype will be present many times amongst the people I'm sequencing. So when I sequence each of these individuals to a few x coverage, any one haplotype that's present more than a few times is going to have sort of an amalgamated coverage that's really quite sensible. So just in this simple example, that light blue haplotype is present 5 times, and so that now gets a coverage of 10x, and by the time I've sequenced 1,000 individuals, then I'm actually going to have hundreds of x coverage of that haplotype, and the green one and the blue one. And so what happens when you sequence many individuals at low coverage is that you don't get any one individual's genomes sort of perfectly, but you can work out what these different types of haplotype or genome are in the population. You've got a very big jigsaw puzzle to put all the information back together, and that's why there's an area of very active research working on the statistical methods for sort of reassembling these individual genomes. But what I hope to convince you of, I think, in the next few minutes is that as an approach, it actually is a remarkably powerful method. Okay, so as I said, all the data is available from these FTP sites, and there's a website to go with it. So I just wanted to give you a sense of what the project has generated. Oops, wrong way. So the first thing is, it's, of course, we've discovered a lot of SNPs, a lot of indels, a lot of structural variants. We've worked very hard to make sure that the quality of the variants that we have discovered is good, but even still, the issues of calling variants from high throughput sequencing data are complicated. The genome is still a difficult thing to sequence in some parts, and so there are false positives in the data. We've limited it to be, I think, of the order of a couple of % or something, but that's still appreciable. So, but this is sort of a snapshot of the SNP variants that we've discovered in the TRIO pilot, the low coverage pilot, and then the exon-centric pilot. And what these Venn diagrams are showing are the numbers of SNPs discovered that are shared, say, in dark here. These are SNPs that we've discovered in each of these 4 populations in the pilot. So there are 3.7 million of them that were previously known, that we found again in every population. This is the set that are novel, the ones that we've found that weren't previously in dbSNP. And you can see the single greatest contribution to novelty comes from the African population, not surprisingly. That's related to what Nigel, I think, was saying earlier about sort of individuals from sort of unstudied populations tend to contribute more variants. But we still find, we're finding 400,000 or so variants, SNP variants, which are common between all populations. And so these are common variants that may influence phenotype, whereas these are going to tend to be rarer variants that are restricted to the individual populations. So it gives you a sense of the sort of what we've found in terms of the absolute numbers of the variants. 15 million variants, and over half of them novel SNPs. But actually just finding lots of variants isn't good enough. What we're aiming to do is to provide a really quite a comprehensive catalogue. We don't want to just say we found lots, we want to say that we found 95% or more. And so what this chart is showing you is the rate of discovery of variants as a function of the frequency in the individuals that we're sequencing. So we're sequencing 180 genomes. This is actually just from the European population, where we've got 60 genomes or 120 haploid genomes. And so what this is saying, if you take the red line on the top here, here's a SNP that we knew was only present where the non-reference allele was only present in 2 copies in the individuals that were sequenced, because we had some external data. In this case, we had the HapMap data. And of those, we rediscovered about 70%. So this is measuring our power to pick up variants at different frequencies in our sample. Now, if you had perfect genome sequencing, this would be a flat line up at the top. What it's saying is that we don't have great power to sort of detect all these rare variants, the things present 1 or 2 times in the population, but by the time there are about 5 or 6 copies of an allele in the individuals we're sequencing, we're up to about 90–95% power. You get slightly different numbers depending on whether you're looking at HapMap as a resource, a reference resource, which is essentially represents the easy part of the genome, or the exon pilot SNPs, which represents sort of a more average snapshot of the genome. But we're still at about 90–95% power for things present about 5 times. So that's saying that we're not just discovering a lot, we're actually providing a pretty comprehensive catalogue. But then one step beyond that is to say it's not good enough just to say, here are the SNPs, or here are the indels, or structural variants. We also want to provide accurate genotypes, because one of the ways in which people are going to use these data is to look at the genotypes and essentially import them into their data, as I'll describe later. So we need to know that our genotype accuracy, so this is the individual genome genotype accuracy, is high. And so what this chart shows you is the accuracy, again, as measured compared to the HapMap project, at sites that we know are homozygous for the reference in red, heterozygous in green, or homozygous for the alternative in allele in blue. This chart just shows you the sort of the number of things at different types that we're comparing it against. And the bottom line is that for sort of rare things where the accuracy is about 90–95% at heterozygous sites, and then for things sort of more in the middle, it's a bit higher, and it drops again a bit towards the end. But the overall our error rate is only 2 or 3%. Now that's an error rate that's higher than conventional SNP genotyping, but remember, each allele on average has about 2 reads that are hitting it. So this is a very, very low amount of data that we're getting on any one individual genotype, but because we can combine information across samples using this idea of there being a sort of a limited number of flavors of haplotypes, then we can actually get really accurate genotype information out of it at the end. And just by comparison, we reckon that the accuracy that you get out of this low coverage sequencing at about 2 or 3x is equivalent to having a single individual's genome sequenced to about 20x or thereabouts. And that accuracy will go up the more individuals that we're collecting information on. So SNPs are in a way the easy part of the genome. As we've heard, structural variation can be a fantastically important aspect of variation, not just in pathological settings where you've got a sort of a strong Mendelian effect, but also in some sort of complex disease settings. And although there's been a lot of work on structural variation detection in the last few years, I think most people would acknowledge that the degree to which we have a comprehensive picture of common structural variation is very low. Unlike SNPs, we knew about 50 to 60% of everything that was out there above a 5% frequency or so before this project. For structural variants, that proportion was much lower. So calling structural variation from high throughput sequencing is not a mature science. I think Nigel said that you use 8 different algorithms to do it. You know, that's kind of the state. It's very hard, and so different algorithms pick up different features, and you get much better set by combining information across different algorithms. But nevertheless, you sort of put it together, and this is the sort of the distribution of the numbers of variants we've detected as a function of their size, and you can see this is roughly geometric distribution. These are the insertions on the right, these are the deletions on the left, and you can see reassuring peaks around allos and lines, insertions and deletions, less clear in the deletions. And this purple line represents the fraction of the things that we found that are novel. And you can see, you know, in these short insertions and deletions, particularly the insertions, the vast majority of what we're finding is novel. So it's still not a complete picture. We estimate that we have about 80% or so power on average for these things. So there's probably 20% of the things in here that we're missing, but that's a huge advance over what was known before. So I've tried to give you a very brief snapshot of what we've got out of the pilot, and I'm afraid it really is very brief. I just want to give you a sense of how complete a picture of genetic variation we expect to have by the end of the project. We can do this by taking, again, comparing what we found in the pilot to these external data sources and trying to extrapolate what fraction of variants at different frequencies in the population we'd expect to get out of sequencing, say, 500 Europeans. That's what's shown in this chart. So I'm just going to try and pick out a point on here, really. So you imagine things that are a frequency of 1% in Europe. Now that's quite hard to pick up. You know, you have to sequence quite a few people to get that reliably. And because populations tend to diverge from each other through genetic drift, something that might be a percent in one population might be 0.1% over here or even 2% over there. So you have to account for that. But if you put that all together and extrapolate to what we expect to get out of sequencing 500 people or 100 each from 5 populations, we expect in the light blue line, wherever it's gone, the green line, that over 95% of things at 1% frequency in Europe will be detected and genotyped accurately in the full project. So that's our expectation. That by the end of this project, anything that's 95% of things that are at a frequency of 1% or more will be documented and accurately genotyped. Okay, so that's what it is, what we hope to achieve in the full project. Now, I want to say just a few things about how we expect people to use the project genotype data. And I think there are 3 things that I want to pick up. I think there are lots of different ways people use it, but I just wanted to highlight 3. So the first is the idea that we can take this reference panel that we've now constructed and use a technique called imputation to essentially in silico genotype the variants that we've discovered in this project into existing studies where we've already got genotype data, say from a SNP shift. So this cartoon just outlines what I'm talking about. In a lovely ideal world, a genome just consists of noughts and ones. These represent the different alleles that you might find at any position. So you can think of the data that we collect largely as noughts and ones. And if you've done a SNP genotyping study, what you'll have used is one of the existing products which genotype 500,000, 1 million or so variants across the genome. And so you'll have genotypes at a subset of these positions. And then you can use a statistical framework. We use one in Oxford called Impute, but there are others available. And what you can do is to try and fill in the sort of the missing holes from your reference panel. It's a sort of a well characterized route to using a reference panel and some additional data that you've collected, and it's actually proven useful in a bunch of genome-wide association studies, and so you can fill them in. And that actually gives you power to assay a lot of new variants without doing any extra work. So people are already doing this, not surprisingly. It's free. Here's an example of a genome-wide association hit, in this case for an expression, an EQTL, an expression quantitative trait locus, where what you can see around this particular gene, there's a whole bunch of SNPs here which have been imputed and which show very strong association with expression. The blue are things from 1,000 genomes. The red are things from actual direct genotyping using a pretty sparse Illumina array. And you can see there's a sort of a jump of, I think that's 5 to 25 or something in minus log 10 p-value. So that's many orders of magnitude increase in the evidence for an association there. Now it turns, so this is very encouraging, and there are actually already reported studies in the literature where people have used imputation and found real effects. It turns out that actually you can't impute very accurately from the 1,000 genomes data at the moment, particularly for rare things, but it kind of turns out that doesn't matter hugely either. It's not so much how accurately you can impute that matters, it's just whether or not you know that variant exists. So as long as you can impute with accuracy of about 60% or so, typically that's enough to pick up, well, that picks up a lot of new associations. Surely there's more out there if we can impute with more accuracy. So that's one way in which people can use it. But this is the way we've heard a bit more about today, the idea that we can screen for functional variants. So the idea is that you've taken your genome, you've sequenced it, you look at the mutations you get, you're interested in selecting out those mutations which have given you some nasty phenotype. And so what you do is you compare your genome to some reference and you screen out all those things that you've seen in that reference. In this case, we've got the 1,000 genomes, so we can essentially just eliminate anything that we see in the 1,000 genomes as being kind of uninteresting. So, question you might like to know the answer to is, if you do that, on average, how many variants would you have left if you were just a normal person without any nasty phenotype? So what these curves are trying to show you is, if you take take that top one here, suppose we had, we'd put all of our 1,000 genomes individuals together. This is in terms of haploid sort of genomes. Suppose that you put them all together, what number of novel variants would you expect an individual to have that weren't in that the 1,000 genomes resource? And this, in this particular case, this line is the number of non-synonymous variants that you would expect to have, the ones that you haven't seen before. And the answer is about 100, 150 or so. Now, the ones on the bottom are perhaps the ones that are more interesting, the ones sort of flatlining along here. These are ones where there's some strong chance of these being sort of functionally important because they essentially destroy the function of the gene in which they landed. They're a premature stop codon or a mutation in a essential splice site. And so you don't have many of these, you only have about 50 to 100 of those. By the time you've got up to, you sort of screened against the 1,000 Genomes Project, the vast majority of those have already, you've seen somewhere else, and so you can essentially eliminate them from your studies. Now it turns out this shows you the absolute number, so by the time you're up here, you're talking a few tens. However, it's in terms of the fraction of everything that you get that's novel. Of course, that's highest for these loss of function variants because these are the things that typically have which have the strongest skew towards rare variants. So in a way, I think that's quite encouraging. It's saying by the time you get out to just this pilot study size, 95% roughly of these loss of function variants you will have already seen. 95% of the loss of function variants I have will be somewhere else in 1,000 genomes, and I can just eliminate them from my inquiries further. And that's a really powerful statement, and that curve will obviously continue to go down as we add in more samples. There's as David had an example like this, is a sort of salutary warning about the quality of the databases that exist out there for which sort of assign function to particular mutations. This is a gene, UNC2A mutations in it cause something nasty called Usher syndrome. In dbSNP, there are 66 missense variants reported. Two-thirds of these we detected in the 1,000 Genomes pilot. There are, in addition to these sort of regular segregating variants, some of these are also the nasty disease-causing ones that you find in OMIM or the Human Genome Mutation Database. Those are the nasty ones that you don't want. And we found some people from the African population which are apparently homozygous for these disease-causing mutations, and these are not compatible with life. And so that's clearly wrong. It's a misannotation in the database. Actually, other reports had previously shown that this isn't a real disease-causing variant. And so I think actually one thing that we're going to learn in the next few years is an awful lot about these databases of disease-causing variants. And there's going to have to be some process of reannotation. Finally, in terms of use, I think an important approach that I've probably given some hints about that is actually really powerful is that genome sequencing isn't really the end of genome SNP genotyping. It actually makes SNP genotyping much more powerful by this approach of imputation that I talked about. So on the one hand, we have the sequencing itself can spur a whole new wave of SNP genotyping, and this I've stolen from the Illumina website. It shows the development of their SNP genotyping platforms, and down here in this, that says 1,000 Genomes Project up there. So in the sort of 1,000 Genomes Project and post world, they're actually introducing new chips which will scan much more of the genome. So that's going to, that's much lower cost, of course, than genome resequencing, but it's going to be very, very powerful for researchers. Okay, in the last 5 minutes, I just want to end with saying something about what the important challenges for population scale sequencing are looking forward from here. Now, we've already heard something about this. Data handling and analysis is a very important issue when we're dealing with the kinds of volume of data that we get out of genome sequencing. And so for that, it's very important to have sort of efficient ways of summarizing data in sort of compact manners. And so one thing the 1,000 Genomes Project has done is to try and establish a series of formats for exchanging data and reporting data, which are sort of information rich and I think are becoming essentially community-wide supported formats. So that's one important thing. But another important thing, and this is particularly important for individuals sort of working in a lab, is that you really don't want to, because there's so much data, it's very easy to get sort of cowed by the nature of it and not try and sort of interrogate it in a sort of simple one-to-one way, you and the reads themselves. And so that can lead to problems, because often if you just stare at a region where there's some genotyping that you've sort of some variants that you've called from the read data, just taking a look at it tells you that there's something funny going on. And so actually something that I don't think is very well developed yet and is an area that really needs some active research is the sort of the data visualization, being able to sort of zoom in on regions that look interesting or look unusual and actually use your own eyes to pull apart what's going on there. And this is just a region with a known deletion showing the reads stacking up, but you can see also something unusual going on. There's a region that seems to have far too many reads stacking up, so it's got too high coverage. So there's some interesting thing going on, and actually just visualizing the data can help give you hypotheses about what's happening there. Finally, in terms of the sort of data handling, actually a very substantial issue is trying to minimize redundant data transport and storage. Data transport is a real issue when you're dealing with cohort size sequencing projects. And that's why things like we've stuck the 1,000 Genomes data in collaboration onto the Amazon Web Services cloud. And we don't know quite how it's going to be used yet, but hopefully people will use it there rather than trying to transfer everything over to their hard drives and blocking up the internet for the next 2 years. Another thing looking forward that's important is a sort of technological and bioinformatic approaches to improve genome accessibility. What I mean by that is that because the human genome is sort of highly repeat rich, and most of the high throughput sequencing has pretty short reads, although that's improving a lot. In the pilot project, we had to exclude about 15% of the genome from our variant calls just because we couldn't access it reliably, and so you can sort of make masks, and this is what this shot shows at the bottom of where we think we could and couldn't call, and you can see it's got this sort of very bitty structure. It's not like a big block, little block. It's really interspersed. So we had to exclude quite a lot. The good news is that you can actually you can make very quick inroads into the remainder of the genome just by increasing the read length. So even if you had 75 base pair paired end reads, we've now 150 base pair is quite accessible, at least in some people's hands. Then most of this sort of 15% actually can now be accessed. So that's kind of really good news, but there is going to be some rather resistant fraction of the genome that is going to be hard to access even with sort of improved read lengths. And that's where the sort of my final challenge arises, is the idea that while most of the approach to genome sequencing has to date focused on the idea of getting a whole bunch of reads and mapping them to a very high quality reference, we know that one reference is a very poor representation of the entire variation that exists in the human genome. And so what we do, what happens is that there's a bit of DNA that I have that's not in the reference. I have some reads from it. They're not going to map when I try and stick them back in the genome. And so what I need to do is to integrate essentially mapping approaches with approaches to analyzing variation which are more like de novo assembly, i.e. sort of trying to reconstruct the genome from scratch. Now you basically can't do full de novo assembly from the short read data in a very substantial way yet, but I would just want to give you an idea of what you might be able to do that is limitedly useful. And this is actually in a context that arises from Matt's talk, which is talking about HLA genotyping from short read data. So the HLA is this very highly polymorphic region on chromosome 6. If you look at reads mapping to the region in any one individual, you'll find big holes, and that's because there's lots of structural variation and there are some very polymorphic exons. And so the conventional mapping approaches don't work well. So what Zam Iqbal has been developing is it's actually a general, it's not just HLA, but this is a sort of an exemplar of its use. He's developed a de Bruijn graph approach, which is a sort of de novo assembly approach. But whereas most de novo assembly methods tend to try and create a consensus genome, Zam's method actually specifically goes out looking for variation that you can see through particular bubble structures in the graph. And I'm not going to go into de Bruijn graphs, but the idea is that you build a graph structure and you look for bubbles in it. How does that help us in the HLA? Well, what we can do is we can take actually all the known, in this case HLA B alleles that are out there. We can build a graph out of those alleles, and then we can sort of thread our own short read data onto that, and to see which subgraph is lit up. So we can ask, can we actually use this short read data in conjunction with the in conjunction with our graph of sort of known HLA-B alleles to pick out what the correct individual's genotype is. Now, that all sounds nice in theory. This is just a picture showing you the graph of all the HLA-B alleles, and you can see that's somewhat messy, and by the time you've included the rest of the genome, that looks significantly more messy. But in this approach, so this chart is trying to show you the likelihood for every possible genotype pair at HLA-B. Here's the best one that you get out of it. That's its particular coding, and this matches the experimental HLA typing result. Things nearby are related, although slightly different, so there's clearly work to do, but this is very encouraging that you can actually sort of integrate what's quite like a mapping approach with a de novo assembly to pull out genotypes for these highly variable regions. Okay, so that's essentially all I wanted to say. Where are we going next? The 1000 Genomes Project has Currently data on over 500 individuals, expect to have more than 1,000 done by the end of the year. This idea of sequencing at low coverage is becoming quite popular, and I'm involved in a study on type 2 diabetes, sort of using the same model to sequence several thousand people who have type 2 diabetes. So it's a model that's going other places, and the data is already proven useful in many medical genetic studies, so this is an advert for people to go off and use it, please. It's a big project, many people to thank, and I've just listed the co-chairs of the various groups involved. Thank you.